This is a new series of lectures on nucleic acid and protein synthesis. It includes the structure of DNA, structure of RNA, DNA replication, RNA transcription, translation or protein synthesis, and mutation. In this context or a series of videos, this is our first lesson, and we'll discuss structure of DNA in this video. So let's see a nucleic acid. Nucleic acid is a substance or a material which has low pH and is found generally in the nucleus of eukaryotic cells. So that is why it is called nucleic acid. There are two types of nucleic acid found in the nuclei of eukaryotic cells. It is DNA and RNA. DNA stands for deoxyribonucleic acid, whereas RNA stands for ribonucleic acid. So let's talk about DNA in this lesson. DNA, as I said, is a nucleic acid, is a type of nucleic acid, and it is a genetic molecule. It can be accurately copied and passed on to daughter cells. It also can store coded information which controls the characters of a cell, or which controls the synthesis of proteins, which control the characteristics of cells. So that is why it is a genetic molecule. DNA is a macromolecule. It has a larger molecular mass, so that is why it is a macromolecule, and is a polymer of nucleotides. So nucleotide is a monomer unit of nucleic acid. It is monomer unit of not only DNA but that of RNA too. So before we discuss structure of DNA, let's talk about the structure of a nucleotide first. Structure of a nucleotide. A nucleotide consists of three components. It has a nitrogen base, it has a pentose sugar, and a phosphate group. Joined together by the covalent bonding. There are two types of nitrogen bases. They can be classified into two groups. One are purine bases and other are pyrimidine bases. Purine bases have double ring structure and pyrimidine have single ring structure. So purine have higher molecular mass, whereas the pyrimidine have lower molecular mass. So you can see there are two types of nitrogen bases, pyrimidine bases and purine bases. You can see the purine bases have a double ring structure, a hexagonal ring and a pentagonal ring that are joined together, whereas pyrimidine bases have the single ring structure. You do not need to know these structures for an A levels. However, you should know that there are 5 different types of nitrogen bases that are named as adenine, which is represented with adenine. It is represented with A. Guanine. It is represented with G. Cytosine, represented with C. Thymine, represented with T, and uracil, represented with U. Adenine, guanine, and cytosine, these three are found in both DNA and RNA. Found in both DNA and RNA. Whereas this is found in DNA only, and this is found in RNA only. The sugars can be classified as monosaccharides and disaccharides. Monosaccharides are further classified into various categories. As we have discussed that in carbohydrates. So monosaccharides may be trioses if they have 3 carbon, tetroses, pentoses, and hexoses. Trioses, as I said, they have 3 carbon, tetroses have 4 carbon, pentoses have 5 carbon, and hexoses have 6 carbon. We have discussed the structure of glucose and fructose in detail. Nucleic acid, however, contains pentoses in them. There are two types of pentoses. One are ribose and other is deoxyribose. So these are pentagonal rings. Each have the 5 carbons, so that is why it is called a pentose sugar. The difference in between the ribose and deoxyribose is that at carbon number 2, you can see they have been numbered as carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. So carbon number 2 has this NOH group here, whereas the carbon number 2 in deoxyribose sugar has a hydrogen instead of hydroxyl group on carbon number 2. So there is one oxygen is less in deoxyribose. The name also suggests the D means removal and oxy from an oxygen, and ribose, a ribose which has one oxygen less at carbon number 2 is deoxyribose sugar. Then a phosphate group. Or a phosphoric acid group. Phosphoric acid group can be written as H3PO4, or this may be P double bond O, OH, OH and NOH. So this is the phosphoric acid group or a phosphate group. Now these three components, as I have drawn the diagrams, and I've talked about nitrogen bases, a pentose sugar and phosphate group. They are covalently bonded together to make a nucleotide. One nitrogen base binds with the sugar on carbon number 1 and a phosphate group on carbon number 5. So let's see how they join together to make a nucleotide. So a nucleotide, you see, this is for instance a deoxyribose sugar, as it has one oxygen less here at carbon number 2. So this is deoxyribose sugar. So a nitrogen base is attached at carbon number 1 here, and this phosphate group is attached with this carbon number 5 here. So this makes a nucleotide. You don't need to know this structure. However, you should know that this nucleotide has a sugar with a nitrogen base on one side and a phosphate group on other side attached with it. We can represent this phosphate group with simply P. So depending upon the nitrogen base attached with the sugar, as there are 5 different types of nitrogen bases, so it has the different types of nucleotides. So let's see the different types of nucleotides. So there are total 8 different types of nucleotides. 4 nucleotides of DNA and 4 nucleotides of RNA. The difference in the DNA and RNA would be the DNA has deoxyribose, ribose sugar, and thiamine here. Instead of uracil, whereas RNA has RNA has ribose sugar instead of ribose sugars. So they would have an OH group on carbon number 2 instead of an H on carbon number 2. The other difference between an RNA nucleotide and the DNA nucleotide is that the RNA nucleotides will have uracil instead of thiamine. Okay, so this is uracil instead of thiamine. So there are two basic differences in the DNA nucleotides and the RNA nucleotides. Number one, DNA nucleotides have deoxyribose sugar and RNA nucleotides have ribose sugar. The other difference is that DNA nucleotides have thiamine, and in RNA nucleotide this thiamine is replaced with uracil. Dinucleotides now. A dinucleotide consists of two nucleotides joined together by a linkage in between them, which is called phosphodiester linkage. So two nucleotides are joined together by condensation by a linkage called phosphodiester linkage. So let's see the formation of a phosphodiester linkage. For instance, we have these two nucleotides. We have this one nucleotide here and we have another nucleotide here. So we have two nucleotides. These two nucleotides are joined together by condensation. This condensation reaction involves a removal of water molecules from carbon number 3 of one nucleotide and hydroxyl group from the phosphate group of the other nucleotide. So a water molecule is eliminated out of it and a bond is formed in between carbon number 5 and carbon number 3 through a phosphate in between them. So this is an ester linkage. You can see there are ester linkages formed here. This is an ester linkage between carbon number 5 and phosphate group here through an oxygen, and there is another with carbon number 3 and phosphate here. So there are two ester linkages by a phosphate group in between them. So that is why this is known as phosphodiester linkage. So this is how two nucleotides are joined together by a phosphodiester linkage. This can be broken down. A dinucleotide can be broken down into two nucleotides by a reverse process, which is known as hydrolysis. So a water molecule will be similarly added, and this can be broken down into the two nucleotides. Enzymes can break down the nucleotides, two nucleotides into individual nucleotides. More nucleotides can be joined repeatedly by many condensations, making a long chain of nucleotides, which is known as a polynucleotide. Polynucleotide chain. So you can see here, if there are two nucleotides joined together, this is a dinucleotide. But if there are many nucleotides joined together, this would make a polynucleotide chain. Before we discuss the structure of DNA, there is an important point that you should consider, that each of this nucleotide in this dinucleotide or a polynucleotide chain has a phosphate group, you see, on one side, and a carbon number 3 on the other side. So this determines the two terminals of this chain. This is 5 terminal or 5 end, and this is 3 end. By now you have understood the structure of a nucleotide, how the two nucleotides join together by phosphodiester linkages and make a dinucleotide or a polynucleotide chain. And a polynucleotide chain has two terminal or two ends. One end, as it is shown here above, that has carbon number 5 of this sugar upwards, and that is why it is 5 prime, and the other is 3 prime. The last nucleotide has the carbon number 3 of the sugar downwards, whereas this first sugar in this chain has, every sugar has the carbon number 5 upwards and carbon number 3 downwards. So that is why it is 5 prime and 3 prime. So after you have understood these, now we are in a better position to understand the structure of DNA. So let's see structure of DNA. A DNA molecule has two polynucleotide chains held anti-parallel together. These two strands, are held together by hydrogen bonding between the specific base pairs, and each polynucleotide is a right-handed helix. So a DNA molecule is known as double helix or duplex or double helical structure. I'll talk about these points one by one. So let's see what does this the two polynucleotide chains held antiparallel together means. In this diagram, you can see one strand that has a 5' upwards and 3' downwards. Okay, whereas the other chain, polynucleotide chain, it has 5' downwards and 3' upwards. Okay. So these two chains are said to be anti-parallel to each other, as the both these strands have their opposite terminals facing each other. One has 5' and the other has 3' upwards, and these two strands are held together by hydrogen bonding in between the specific base pairs. So this is the hydrogen bonding in between the nitrogen bases. This hydrogen bonding is specific in between the base pairs. Adenine always pairs with thymine. Cytosine always pairs with guanine. Thymine pairs with adenine. There are two hydrogen bonds between thymine and adenine, and there are 3 hydrogen bonds between cytosine and guanine. So this has 2 hydrogen bonds. So this base pairing is specific in between these 2 nitrogen bases. The third thing that we are going to discuss is that each polynucleotide is a right-handed helix. And so a DNA molecule has a double helical structure, or it is a double helix. So this finally shows the double helical structure of a DNA molecule. If this is, for instance, 5' end of 5' end of this strand, this will have 3' and on this side. The other has 5' on this and 3' on this. So these two strands are held anti-parallel to each other. And each is a right-handed coil or right-handed helix. And these two strands are held together by hydrogen bonding in between the specific base pairs. So if this one strand has adenine here, guanine here, thymine here, and cytosine here, the other strand has complementary bases. Thymine is complementary to adenine, cytosine is complementary to guanine, and adenine is complementary to thymine, and guanine is complementary to. So cytosine and guanine are complementary bases, and thymine and adenine are complementary bases. So there is hydrogen bonding in between the complementary bases. This hydrogen bonding is shown with the dotted lines here. As I told you about this specific base pairing, showing there are 2 hydrogen bonds in between adenine and thymine, and there are 3 hydrogen bonds in between guanine and cytosine, and 2 hydrogen bonds in between adenine and thymine. So this is the double helical structure of DNA. Now let's see, we can use this concept to calculate the total number of hydrogen bonds in a DNA molecule. You can have numerical problems in your MCQs paper, or maybe in paper 2, that require you to calculate the total number of hydrogen bonds in a DNA molecule. For instance, you have a DNA molecule shown here in this diagram, which has 20 bases or 20 nucleotides. A DNA molecule has 20 different bases. %25 of it is adenine. Calculate total hydrogen bonds in this DNA molecule. Also state to calculate the total number of hydrogen bonds in this DNA molecule, as there are 20 different bases, so 25% of it is adenine base. So you can have 25 divided by 100 multiplied by 20. So you will have 20 this 100 can be cancelled out by 20 by 5 times, and this 5 that cancels out, so this 25, so you will have 5 here. So there are 5 adenine bases. As it is a complementary base pairing, so adenine makes double bonding with thymine. So if there are 5 adenine bases, there must be 5 thymine bases. So there must be double bonding in between them. So we will multiply 2 into 5, that would give us 10 hydrogen bonds. As there are total 20 different bases, so 5 are adenine bases and 5 are thymine bases. So the remaining are adenine bases plus thymine bases is equal to 10. So guanine bases plus cytosine bases will be equal to 20 minus 10, so that will be 10. So as it is a complementary base pairing in between guanine and cytosine, so half of them would be thymine and half of them will be guanine and half of them will be cytosine. So as it is total 10 nucleotides that have guanine and cytosine, so 5 guanine will have triple hydrogen bonding with 5 cytosine, And to calculate the total number of bonds, we will multiply 3 with 5, and that would give us 15. So this would give us 15 hydrogen bonds. The total bonds formed will be this 10 and 15 is added up together. So this would make 25 hydrogen bonds in this DNA molecule. So this is how you can use this formula to calculate the hydrogen bonds in any of the DNA molecule. Now, to make sure you have understood this and have calculated it correctly, you can see you can also count the number of hydrogen bonds in this DNA molecule, as it is a very small molecule. So you can count the number here. So this has 3 here, this has 2 here, this has 2 here, this has 2 here, this has 3 here, this has 3 here, this has 3 here, 2 here, 3 here, and 2 here. So if you count them together, so that would make 3, 2, 5, 2, 7, 2, 9, and 3, 12, 3, 15, 3, 18, and 2, 20, 3, 23, and 2, 25. So this is 25 hydrogen bonds that we have calculated in this way. So as it is a very small molecule, you can easily count the number of hydrogen bonds that are shown in this diagram. But if a DNA molecule has not been drawn and it is a very long DNA molecule, then you would have to use this method to calculate the total number of hydrogen bonds, minimum number of hydrogen bonds, or the maximum number of hydrogen bonds.